Today I wanted to discuss sex and strain differences in the natural history of the SCN8A-N1768D mouse model. And at the very end, I want to discuss with you some of the new mouse lines that we've made using this model that I would love to share with the community. So the purpose of our study is to observe the entire life history of the mouse with regard to seizure onset, seizure activity patterns, sleep comorbidity, and mode of death. And we do this by 24/7 video recording. Ultimately, we'd like to establish a baseline for off-target drug testing using this baseline of the natural history of the untreated mouse. And ultimately, we would also like to define critical points in the life history in order to intervene and take the hippocampus and sequence RNA transcripts, do pathway analysis, and identify novel drug targets before and after seizure onset. The model and the system we're using basically is this. We follow the mice, and at various time points before seizures begin, we take samples of the hippocampus, and by observation, we know exactly when their first seizure occurs. So we take samples immediately after the first seizure, and then we let other mice progress on to additional seizures and sample them at various seizure points. We compare the heterozygous mice that are undergoing these seizures with their wild-type litter mates, or we compare the homozygous mutant mice, we call mutes, with their wild-type litter mates as well. And that's what we do, and we get the tissue, and we do sequence 23,000 gene transcripts from the hippocampus. This shows you a single mouse, the life history of a single mouse from the time it had its first seizure at the onset point, and then the series of seizure bouts that it had on this axis, the number of seizures, and these are tonic-clonic seizures. And then we define these periods of 3 or more days as gaps. And there's seizures, bouts, and seizure gaps. And there's a series of bouts and gaps. This is very typical for our mice. And at the very end, here you see the final seizure bout and a terminal tonic seizure. And what we measure when we look at these mice is the age at which they perish, the number of TCs they've had in their entire life, their age at the first tonic-clonic seizures, and the number of days they survive after that. We analyze the gaps and the bouts, and we look at the mode of death, and I will describe 3 different modes of death. We look at sleep architecture, and we count the number of seizures in the wake and sleep period. And just to say that we've now followed over 14 mice on average per day for a year and observed over 5,000 tonic-clonic seizures. So what I'm going to present to you today will encompass some of what we've learned. This basically shows you a table of the mice that I want to report to you now. For simplicity, we're going to limit our study to just virgin females and virgin males. And these 21 mice were followed for their entire lifespan, 12 females and 9 males. The total number of tonic-clonic seizures, you can see, we looked at over 1,500 tonic-clonic seizures, many more in females. The females also survive longer, about 130 days versus 100 days for males, significantly different. And you can see that females have many more seizures, about 100 versus 50 for males. The number of seizures per day is also about 0.72 versus 0.42 for females versus males. The age at first seizure onset is similar between the two, but you can see females survive for many more days beyond their first seizure. And the gap analysis is interesting as well. Females will have more gaps and slightly more bouts. But ultimately, what we're discovering is that the females are much more resistant to the effects of seizures, despite the fact that they have more seizures, they live longer and survive better. This is showing you a Kaplan-Meier curve for survival for males and females, the males in red and the females in blue. And you can see that males start to die off sooner. But the rate of death is similar to the females. The females seem to have a prolonged period of survival before they start to perish. And you can see on the bottom part of the figure, the accumulated number of tonic-clonic seizures on average that each has. So males, like I said, will have fewer seizures in their lifespan, whereas females will continue to have many, many more seizures and are more resistant to them and survive longer. If we look at the number of seizures by the hour of the day, males and females are quite similar. You have males and females having more seizures in the light cycle. And the light cycle is when the mice are sleeping. And then in the dark cycle, the mice are more active, and you can see they have fewer seizures. There's some slight differences in these patterns, but overall they're similar. And if you look at the number of seizures that occur while the mice are sleeping or waking in in activity, you can see that there's many more seizures. Roughly between 65 and 90% of the seizures occur in the sleep stage or upon the sleep-wake transition, whereas a smaller number of seizures occur when the mice are active. And that's true for both males and females. Now I want to talk a little about mode of death. We have three different modes of death. SUDEP we define as a few seizures on a day when there were very few seizures preceding those seizures, either on that day or the day before. In other words, a seizure-free period followed by 1 or 2 seizures and death. We also have observed a pattern where females, or both mice, but mainly females, will have a flurry of seizures that they have at shorter and shorter intervals, and they can have up to 20, 30, 40 seizures in these periods of clusters, and seem to just run out of steam and die at the end of this. So we just call that convulsive status epilepticus. And finally, we've discovered another mechanism of death, another mode of death that is almost entirely in males, we call decompensation. And basically, the males will have had a series of seizures, but then they have a couple of seizure-free days and slowly start to die. They lose their appetite and thirst, then the respiratory tone declines over a period of 24 to 48 hours. And what I'm showing at the top is the breath rate in hertz in the piezo cages we use to look at sleep, and you can see the slow decline in respiratory rate until the point of death here. And if you just count the number of these different modes of death, you can see females and males are extremely different. Both have very rare cases of SUDEP. The females predominantly have a convulsive status mode of death, and the males predominantly have a decompensation mode of death. And we don't know exactly what the underlying cause of that is. Now this shows you a slide, again, showing the seizure bouts and gaps for another mouse, and the way we've measured sleep in the piezo cages, where you can see that what I'm showing here on the dotted lines are the longer sleep bouts that are longer than 32 minutes in both the light and dark phase. And what you notice is, normally before seizures begin, you'll have very long sleep bouts, which is healthy sleep architecture. And upon seizure onset, you'll see a great loss of these longer sleep bouts. The sleep architecture is disturbed. And every time there's a sleep bout, there is a loss of these longer bouts. Every time there's a seizure bout, there's a loss of these longer sleep bouts and some recovery of the sleep, but in these gaps. But you can see that seizures disrupt sleep, and to the extent that disrupted sleep leads to seizures, we're not quite sure. But it looks like seizures are the leading edge of what's happening here, and it certainly fits the idea that seizures and sleep are comorbid outcomes of this disease. If we look at males and females in the overall sleep and this longer sleep bouts before and after seizures, you can see that males and females are quite similar. And what I'm showing you here is that these longer sleep bouts are lost during seizures after seizure onset, and there's a gain of these shorter sleep bouts, and it's very similar for males and females. I wouldn't say there's much difference at all. So they're both affected similarly by seizure onset. The sleep architecture is disrupted. What we notice for our females is very interesting. We have, normally females will undergo 4 phases of the estrus cycle: metestrus, diestrus, proestrus, and estrus. And during these different phases, you'll have different levels of hormone. You'll have progesterone secreted during metestrus and diestrus, dying off slightly and increasing in proestrus, and then dying off during estrus. And you have estradiol showing a similar pattern, but really peaking mostly at proestrus and then suddenly dying off. And we know from the literature that progesterone is an anti-convulsant effect, and estradiol has a pro-convulsant effect, which fits nicely with the fact that during the day activity, or during the period of sleep, during the daylight, you can see that females have a peak of seizure activity during the proestrus, which correlates with both the higher estrus secretion and the withdrawal of the progesterone. And progesterone, again, being anti-convulsant, and some have speculated it's this progesterone withdrawal that can lead to seizure onset. And then this shows you a female, the life history of a female, where we were looking at this estrus cycles, and you can see they cycle very nicely in the pre-seizure onset period. So you can see the pro-estrus estra, med-estrus, diestrus. Sometimes it's two days of diestrus. And for this particular mouse, we recorded 11 straight cycles of this PEMDD cycling. And when the cycling stopped, it was predictive of the onset of seizures. So you can see on day 53, the cycling stopped. The mouse got into an odd pattern of pro, meta, and di, and then basically got stuck in diestrus for all of these sleep bouts and gaps, and was not able to recover and died on the final day. What's interesting is that dams, the females that have litters, the survival correlates with the age of breeding. So if you look at the age of breeding, if you have your first litter at 45 days, you're going to die at a sooner day than if you have your litter at 65 days. And that's a correlation with a high correlation coefficient. And so we think that what the hormonal changes that are happening during pregnancy are actually promoting a longer survival. So the females will buy an extra 30 or 40 days because during pregnancy, which lasts about 20 some days, there is suppressed estradiol and increased progesterone expression. So we think that might be part of the explanation for this longer living period in female dams. Virgin females who are housed together will often exhibit pseudo-pregnancy you'll see them humping each other, which is a sign that they're undergoing pseudo-pregnancy and probably expressing progesterone during those periods as well. So basically, we've come up with a simple model that we would like to test. You can see that what we're saying here in the male and female is testosterone is produced in the male gonad, and in the brain, it's converted by P450 aromatase into estradiol. And males in early development express a higher number of estrogen receptors in the hippocampus, higher than females. So males are essentially getting a pretty strong hit of estradiol in the brain, in the hippocampus in particular, whereas females have both estradiol and progesterone expressed in the gonad, and no necessary loss of progesterone. So the progesterone is anticonvulsive, the estradiol is seizure promotive, and we think this might be part of the explanation for the different lengths of survival and the effects of seizures on males and females. Something is protecting the females and not protecting the males. We would definitely like to test this hypothesis, perhaps by looking at aromatase inhibitors in males and seeing if we can create the situation that you see in females. One final thing I just want to mention, which is really interesting, because we looked at enough mice over a period of a year, and we noticed that in periods of time when the barometric pressure changed in Tucson, Arizona, that's the times when mice underwent more seizures. So you can see seizure frequency down here on the bottom across all the mice we were watching at the time. You can see the barometric pressure changes, and these sharp changes occur when a storm comes in. This is the amount of rain. You can see the barometric pressure drops, sharply rises, and this is a time when mice will generally be more seizure prone. And when we actually looked at the 42-week period where we looked at maximum minus minimum barometric pressure over the week, and we plotted that against the number of TCs, tonic-clonic seizures per individual, we had found a fairly significant correlation. Quite interesting. Barometric pressure does affect seizures in mice, and I understand that this is true in humans as well. So I just wanted to end by advertising some of the resources we have that I would love to share with other researchers who are interested. We made crosses between our Black 6 mice and C3H-HEJ mice, and then at various stages along the way, we phenotyped them, and we discovered very different outcomes of these mice as they became more and more C3H genetically. We noticed that, for example, in our N6s, we created short-living and long-living homozygous mutants. So I'll show you that on the next slide. Whereas then we continued crossing to C3H, we lost that phenotype, but we created a phenotype in which the heterozygotes didn't die and don't get seizures. So we created a whole different assortment of lines. Black 6 is shown here in red. These are females. And you can see our N6 with the intermediate survival of heterozygotes. The N10s survive perfectly without seizures. And the N7s and N8s die faster than even the black 6. This is in the females. In the males, you can see that the males actually survive longer than the females in the N7 and N8 lines. And the N6 don't live quite as long in hets males, but that N10 is steady in not having seizures that we can detect and living for at least 6 months or longer without any problems coming from seizures. On the homozygous mutants, the N6s, we have two different strains of N6. Something is segregating in N6, so we've got these long-living homozygous mutants that live up to 90 days. And you can see we also have in this group here some N6 that die at 21 days, just like the N7Bs, the N8Bs, and the N10s. And the black 6 live a little bit longer. We lost that 21-day phenotype in our black 6. So we've constructed these different lines with very different phenotypes. The black 6 that have the homozygous mutants that die at 21 days generally die of a cluster of seizures, convulsive status. However, our N10s, they don't die from convulsive status. They get hind limb ataxia, or hind limb paralysis, which is an observation Miriam Meissler's lab had made earlier in a loss of function SCN8 homozygote having a paralysis phenotype. And we have that phenotype as well as the convulsive status phenotype in the homozygous mutants. And then we have these very different sorts of lengths of survival for the heterozygotes. And I would love to be able to make these lines available to people who want to study them in any way that suits your needs, and what you generally do in your lab. Would be wonderful to learn more about what's going on with these mice. We're sequencing the genomes of some of these strains, and we're doing the transcriptomes, so that'll all be fully described as well. And just to give you a teaser of our transcriptome results, I obviously don't have time to go into them, but we've noticed that the pre-seizure mice have a lot of deactivation of a bunch of pathways that involve metabolism and mitochondrial function, whereas after the first seizure, a whole different set of pathways get activated. You can see the red versus the blue. This is a set of pathways that are getting activated that have more to do with inflammation and cellular remodeling. And we're digging into that. So the next time I give a talk, I'm going to dig into these RNA-seq results and the kinds of pathways that are changing in the pre-seizure and post-seizure stages. And with that, I would just like to acknowledge the students in my lab that have done all this work, and the Acute Syndrome Foundation for putting on this presentation and for providing support for our RNA-seq analysis. And thank you very much for listening.